Hello. Today, our lecture is about genetics. We'll deal with 6 topics. For Mendelian inheritance, we'll deal with the morphological and the molecular phenotypes. The second topic will be segregation of a single gene. The third will be segregation of 2 or more genes, while the fourth topic is human pedigree analysis. And the fifth topic will have some quick review on the non-Mendelian inheritance. And finally, we'll just give a brief, just one slide about genetics and what is genetics today. The main reference for the lecture will be the 8th edition of Genetics by Hartle. And actually, it's not all the book, it's only chapter 3. For the first topic, so the study begins with the famous scientist Gregor Mendel, who grew more than 34,000 pea plants over an 8 years period, including more than almost 6,400 plants in 1 year alone. And from that time, the expression of Mendelian genetics or transmission genetics have been coined. Mendel have found that the morphological phenotypes is around 7 traits or characteristics in the garden bee. So you can see here there are 7 phenotypes, including the shape of the seed, the color of the seed, the color of the flower, the bud shape, and the bud color, and the flower position as well, the flower and the bud position on the stem, and the stem lengths as well. You can see some of them are short, some of them are long. So you can see that there are some characteristics that is dominant over a recessive one. And in this figure, it shows you which characteristic is the dominant and which one is the recessive. Taking, for instance, for the seed shape, you can see that round is dominant over wrinkled. So that's the phenotype when they are crossed. And we will go in details about how he crossed Mendel, how he crossed, and how he managed and succeed to generate the laws of the genetics. So let's see how the molecular basis of the genetics and how they interpret the phenotype into a genotype by this example. So actually, wrinkled seed character of the pea is caused by a part of a gene, which is called the transposon gene, which encode a protein or enzyme called the starch branching enzyme 1, SPEI. So in the wild type, represented by big W, which give the round appearance, this allele give rise to that enzyme. And you can see this is a homologous pair of chromosome, and you can see the location of the gene on the DNA duplex, and this is the wild type. And when they done amplification for that gene and ran this gene on the agarose, they found that it gives agarose gel electrophoresis. They found that it gives a specific band here of that size. While for the wrinkled gene, for the wrinkled allele, actually it is a mutant allele that encode another form of SPE1, which is inactive, because its DNA sequence is interrupted, as you can see from the figure. So this actually the normal gene, once interrupted by another transposable element that jumps inside this gene, it will interrupt the coding of that gene, resulting to inactive enzyme. And when they cloned this and amplified this gene sequence, it appeared on the gel electrophoresis in a higher size, which reflect the insertion of additional nucleotide sequence. So that's why it migrate slowly and appear in higher size than the wild type. In case if the garden pea have like the heterozygous form, in which the morphological is heterozygous in case of W capital and W small, this of course will give the phenotype of round seeds, because as it was discovered by Mendel that the allele wild type always in that case, dominant over the wrinkled type. So you can see when they done the molecular analysis for the heterozygous version of that P seed, they found that it contained the two versions, one for the wild type and one for the mutant type. So because garden bee are sexual organisms, so each cross can be performed in two ways, depending on which phenotype is present in the female parent and which in the male parent. So you can see that Mendel was the first to demonstrate the following important principle, which is the outcome of a genetic cross does not depend on which trait is present in male and which is present in female. So he tried something very interesting, which is called reciprocal crosses. And actually, when he tried these reciprocal crosses, he found actually that the result is the same. As you can see here, he initially made the first cross a female which is homozygous dominant with a male that is homozygous recessive. So basically, this is a round seed, and this is a wrinkled seed because it's homozygous recessive. The resultant offspring was heterozygous, and of course, all of them are round. When he done the reciprocal crosses, in this case, he used the female to be homozygous recessive and the male to be homozygous dominant. The same result appear as well. That means the reciprocal crosses, whatever the trait is on any sex of this, the product actually is the same. So the homozygous have the band which is smaller in size, while the homozygous recessive have the band which is higher in size. Once there is a mating between these two parents, it results, as I said, as a heterozygous individual that have both bands of the gene. So when self-mating of this heterozygous individual, it gave us two types of gametes. One gamete is the dominant one, and the other gamete is the recessive one, same, because both parents are the same. So that gave the ratio of 1 to the ratio of 3 to 1. So you can see here that half of the progeny or the offspring were heterozygous, while quarter was homozygous dominant, while the other quarter were homozygous recessive. So that's mean before that, you must know that it is easier to use this way of sorting, which is called the Punnett square, which provides the graphical calculation of gametes and the progeny. So you can see here, once you merge the dominant gamete, this one, the W capital is W capital, it will give rise to homozygous dominant, the W capital with W small, homozygous heterozygous, and so on. And soon after that, you realize that in F2 generation, you can see that with respect to the phenotype, you have a ratio of 3 to 1. But with respect to the genotype, you have a ratio of 1 to 2 to 1. Why it's 1 to 2 to 1 with respect to the genotype? Because actually the first one is the homozygous dominant, the last one is the homozygous recessive, and you have 2 which is heterozygous. So basically, Mendel have done a lot of experiment with a lot of different combination between the different traits of the P. And in all of them, with all these different mating and this different crossing, it came up the same ratio of around 3 to 1 ratio. And this conclude that there is a dominant character trait and the recessive trait, and always dominant prevail over the recessive in a ratio of 3 to 1 phenotypically. So the key feature of this single gene inheritance, the genes come in pairs. Each cell has 2 copies, which is alleles as well of each gene, dominant and recessive. And for each pair of gene, the allele may be identical, homozygous dominant or homozygous recessive, or different, which is heterozygous. Each gamete contains only 1 allele of each gene, and any gamete is equal likely to include either allele. So there is no preferential in the gametes. So it's like a random process. So the union of the male and the female gamete, as I just said, is random, and they reunite the alleles coming from the parent genotype. And as we said, the reciprocal crosses give the same result, either irrespective of the male and the female, because all trait is not sex-linked. So once the trait is not sex-linked, so the reciprocal crosses will give the same results. So here, Mendel coined the first law, which is the principle of segregation, or it can be called the law of equal segregation. So in this law, he coined out that in the formation of gametes, the paired heredity determinant, which actually the allele, is separate or segregate to give a gamete in such way that each gamete is equal likely to contain either member of the pair. So the gamete can have the randomly and likely the same proportion or percentage to have any of the two alleles. So that was the first law of Mendel. So let's verify this segregation. So there is two types of tests that Mendel do to verify if his theory is working or not. So actually, what's Testcross? Testcross, actually, he mated a dominant phenotype, which in our example could be the round seed, with a homozygous recessive, which is the wrinkled seed, for instance. So the phenotype of the progeny that it have upon this mating will reveal the relative frequencies of the different gamete produced by the heterozygous parent, because the recessive parent contribute only recessive alleles, because if it's a homozygous recessive, it will give only one gamete, one type of gamete. But if it's a heterozygous parent, it will give two types of gametes. So let's see what's happening. So in this case, he took a dominant phenotype. In this case, you can see here, this is a dominant phenotype because it's have the round seed, and he mated it with a homozygous recessive, which have only the recessive trait. And in this mated, he found that it gave the ratio of the offspring to be 1:1. So that's mean in this Testcross, the phenotype, half of the generation will be have a wild type appearance, and half of the generation will be a recessive wrinkled appearance. So what if the parent was homozygous dominant? So actually these kind of tests are done when you don't know the genotype of the parent. So in case if the genotype was heterozygous, you will find that the progeny will have 1:1 ratio. But if the parent was homozygous dominant, in this case, 100% of the 100% of the progeny will be of a dominant phenotype. So these testicles basically give a clue on the genotype of the parent, but you need to do the crossing with the homozygous recessive, because for that you are sure that the homozygous recessive will give rise to only one allele. So that's the Testcross. What about the backcrossing? So backcrossing, actually, you take a hybrid organism that you don't know the genotype of it, and you mate it with a parental genotype. The aim of the backcross, which is sometimes called inbreeding, is to achieve an offspring with a genetic identity which is closer to the parent. It is used in horticulture, animal breeding, and in production of the gene knockout organisms. So what's happening in this backcrossing? So for the original cross, you take the hybrid organism that you need, in this case, to make it pure, and for original cross, you cross it with a parent, which we denoted here like R. So the first backcross will result in organism B, we call it F1 generation. And this theoretically will have 50% of the genes of R. So you do the first backcross with mating with the same parent, you will get another organism which have more into its genome from the R parent. After several backcrosses, you can reach to organism F that have almost 97% of the genes of the parent R, this upon the 5th backcross. Upon the final backcross, you take the organism, the offspring, which is now called BC4 or backcross 4, and you do self-crossing for the aim to obtain an organism that is homozygous for R or nearly as the parent. So in this simplified table, it shows the difference between the Testcross versus the backcrossing. And it simplifies all the info we have said in the Testcross and in the backcrossing, okay? So let's talk about the third topic, which is the segregation of 2 or more genes. So basically, for the segregation of one gene, we talked only about one gene with 2 alleles. In this case, we talk about 2 genes together, how they segregate, how many gametes they produce. So you can see here, actually, you have now 2 traits, the round and the yellow phenotype against crossed with the wrinkled and the green phenotype. And from this figure, you can see that round is dominant over wrinkled and yellow is dominant over green, okay? So this is actually the wild type phenotype, and this is the mutant wild type. So in this case, the type of gamete here, actually, it takes 1 allele from each gene. So you have a WG all capital. And as well, you take 1 allele from each gene, you have W and G small. So the F1 will have this genotype. And actually, because that round and yellow is a dominant trait, so basically it will prevail over the recessive allele. So you have in this double heterozygous generation, you will have the appearance or the phenotype of round and yellow. So what happens if we self-mate or self-cross this heterozygous, double heterozygous individual? So you can see in this case, the story is a little bit different, because when you self-mate, you have 4 different gametes. You can see the combination of the 4 gametes. Basically, the W capital, it have the probability to take in the gamete either G capital or G small for the W capital. And for the W small as well, it have the probability to take in the gamete G capital and G small. So we have 4 different gametes for each parent. So when the probability occur for these 4 different gametes, you will have around 16 individual. And if you categorize this 16 individual, you will have that 9 of them will have round yellow, 3 of them will have round green phenotype, and the 3 of them will have wrinkled yellow, and only 1/16 will have a wrinkled green. So actually 1 will show the 2 recessive phenotype, as you can see here, this one. It shows the homozygous recessive trait and the homozygous recessive for both the shape and the color of the seed. So in this case, if you want to summarize the ratio of this dihybrid cross, you will see that there is 9:3 to 3:1. So that means, although there are 2 genotypes here, which is actually the shape and the color, but both of them segregate or pass into the next generation independent of each other, because each character appear by its own self. So that's why the second law of Mendel talks about independent assortment. Although there is a lot of number of genes, but each of them behave differently. And when they are combined, they'll give a ratio of 9:3 to 3:1. So again, here to elaborate more on how gametes are formed, so we have initially the double heterozygous individual. When it gives the gamete, you have W, which it takes either in the gamete, takes either G capital or G small. So you have quarter the number of gametes will be like this, and the quarter number of gametes will be like this. The other probability, you have the W small, and as well it has the same probability of taking G capital or G small. So that's why it will give you 4 different gametes, and these all the possible outcomes of the probability of the gametes, and all of them have an equal frequency. So basically, if you take one gamete from a bag that contain these lot gametes, all of them have the same probability. You probably will not know which gamete have which genotype. So Mendel actually carried out, he elaborated more and go for a 3 gene cross, And the result of the phenotype confirm well with the ratio of the 9:3 to 3:1. And it gave Mendel another ratio, it's 27 to 9,99331, expected from the independent. And that proof as well that even if there is a lot of number of genes, each gene segregate or pass to the next generation separately, and it doesn't depend on the other gene. And once it's combined in the progeny or in the generation, the one with the dominant will prevail over the recessive. So he coined out the second law, which states that the segregation of the members of any pair of alleles, any pair, 1 pair, 2 pair, 3 pair, even more, is independent of the segregation of the other pair in the formation of the gamete. So that means each pair segregates by itself. So let's do a test cross as well to confirm what's happening for the second law. So actually, in other words, and what Mendel mean in his second law, the genes are unlinked together, unlinked to each other. They segregate separate of each other. So genes that show this independent assortment or segregation are said to be unlinked. So the hypothesis of this independent assortment can be tested directly in a test cross with the double homozygous recessive. So keep in mind, always a test cross is done with a homozygous recessive. Whatever these homozygous recessive come in 1 gene pair or 2 gene pair. So you can see here is a double heterozygous parent is crossed, test cross with a homozygous recessive. So the type of gametes here will be only WG small, while the type of gamete, as we have just said, for the double heterozygous, you have 4 types of gametes. So in this case, you will have 4 different phenotypes in the generation that represent a ratio of 1 to 1 to 1 to 1. So from this generation, you can conclude that the other parent is double heterozygous. So basically, the test cross is done to know and to identify the genotype of the parent, because you already know the genotype of the homozygous recessive, because it will never appear this recessive or mutant trait unless the genotype is homozygous recessive. So once you get a ratio of 1 to 1 to 1 to 1, so that means that the parent, this one, is for sure a double heterozygous. Because, for instance, if this parent was double dominant homozygous, so basically you will have the ratio half to half. You will not have 4 different phenotypes. And of course, you can train yourself doing a test cross with different genotypes, and if you compare the generation generated from crossing these different genotype with the same homozygous recessive, you will have different phenotypes for this generation, which will reflect the genotype of that parent. And this is a summary that shows the difference between Mendel's first law and Mendel's second law. And of course, you can see that the major difference is this one is 3:1 ratio, and this one is 9 to 3 to 3 to 1. And as well, this is a monohybrid cross because it's only involving one gene with the 2 alleles, and this uses a dihybrid cross because it involves 2 genes, each of them is with its own allele. So for the first topic, we'll talk about the human pedigree analysis. So before going into the human pedigree analysis, we need to understand what is the symbols that may appear in this pedigree analysis and what is actually the pedigree analysis. So a pedigree, actually, it's a diagram of family tree showing phenotypes, sexes, and the relationship individual of multiple generation. So in this figure, you can see the circles are always for normal females, the circle, the empty circles, the empty squares for normal male, and this rhombus for the gender unspecified. So that's mean if someone is drawing this pedigree and actually his sex is unknown or deceased, whatever. Okay, so for the other symbols, if it's red in these cases or blocked, so that's mean affected female, and as well affected the male, and the affected gender unspecified. So in this case, if you have a symbol that is like half filled, that's mean it is heterozygous for the allele, and as well for male and female. If you have this symbol in the pedigree diagram, that's mean there was an apportion in this family. And if you have this symbol, that's mean you have a diseased member in this tree. So in this case, you can see from the figure, these are called sibling because actually they are brothers and sisters. And these represent the number of generations. So that's the first generation, the parents, then this is the second generation. And as well, the order is important because the first one is the firstborn and the last one is the lastborn. You can see as well that twins can be represented either monozygotic twins or a dizygotic twins. If there is a mating between relatives, it is represented by double lines, and normal mating represented by single lines. So basically, this slide describes the symbol or conventional symbols of pedigree analysis. So let's see an example of how the autosomal dominant inheritance occur, and have an example on this pedigree analysis. So the autosomal dominant inheritance, what's the meaning of autosomal dominant inheritance? That's mean it's a trait that is autosomal, that exists on our autosomal chromosomes, and it always appear because it's a dominant. So this is seen in homozygous dominant or heterozygous individual because the dominant allele prevail over the recessive. And this diseased individual are present in every generation. So if there is a disease and we don't know if it's a dominant and a recessive, and from the pedigree analysis, we notice that it exists in every generation, that gives a clue that this disease is a dominant. The affected offspring must have one affected parents, and males and females are both equally affected, and this as well indicates it's autosomal and not sex-linked, okay? Because both of male and females are equally affected. On average, half of the individual in the siblings with an affected parents are always affected. 50% of the offspring are affected. The famous example in this autosomal dominant inheritance is Huntington disease. And actually, Huntington disease is caused by a dominant mutation. So it leads, of course, to progressive nerve degeneration that begins at middle age and results into physical and mental disability followed by death. So the dominant allele of Huntington is denoted like HD, that cause the Huntington is very rare. And that's very good for us because it's rare. Because if it's not rare, it will lead to the death of the individual, a lot of individuals. So all affected persons in this pedigree have a heterozygous genotype, okay? That means they are carrier. Heterozygous genotype, that means they are affected. Whereas non-affected person have a the non-affected ones have a normal homozygous with the genotype HD small. So Huntington disease is characterized by being called a complete penetrance. What's the meaning of complete penetrance? That means that the trait is expressed in 100% of the person with that genotype. And of course, because it's a dominant. And you can see from the pedigree here, there is a male parent which is affected, and you can see all the offspring. They have, this couple have 3 children, 2 females and 1 male, and all of them are affected. Once they mate, the 3rd generation as well, it will appear some of them are affected, because of course their genotype now for these siblings, their genotype is heterozygous. Their genotype is heterozygous. So here you can see that not all the siblings or not all the generation is affected. So that shows that the dominant, the autosomal dominant inheritance is represented in every generation. So the non-affected person must have an HD small, because HD is recessive, while the affected person have a genotype of HD allele, at least have one allele that is capital. And of course, it's very rare, and that's good for us. The second example, the autosomal recessive inheritance. So actually this seen in homozygous recessive individuals, and as well females and males are equally likely be affected because it's autosomal, not sex-linked. And the affected individual, if they reproduce with a normal person, usually having unaffected progeny, the mating of 2 heterozygous, which in these cases it's called carriers because they have a dominant and the recessive, and this recessive never gives its phenotype because it is masked by the dominant character, the dominant allele. So it is required to produce an affected that in case only affected person appear in case of homozygous recessive. So most affected individuals have unaffected parents, and the parents of the affected individuals are often relatives. Among siblings of affected individual, the proportion is approximately 25%. And the famous example for that is albinism. So albinism, actually, it is the absence of pigments in the skin, hair, iris of the eye. Another important feature which increase the risk of this autosomal recessive inheritance is the consanguinity mating. Consanguinity mating, this increases the risk. So a mating between relatives increases the probability of having a homozygous recessive descendant, because maybe these relatives, one of them is carrier and the other one is carrier, and when they mate, they give the recessive allele could be combined in the child and give this autosomal recessive inheritance. So this recessive allele usually is hidden in the status of being heterozygous. And this hidden recessive allele can become homozygous if there is inbreeding, which actually in our case as a human, it's called consanguinity mating. And you can see here the pedigree of the albinism. You can see that although the parent appear non-affected, but you can know that they are heterozygous for the character because it appears in their generation. Okay, it appears as it's a carrier in this generation, and in the 3rd generation, the condition appears in almost like 50% of the children. So you can see here how the mating between first cousin resulted to a homozygous recessive, which is affected and increase the risk of appearance of this albinism condition. Okay, so what is this? This is actually the family tree of hemophilia A. It's a condition that affected the clotting behavior of the blood in the royal family of Europe, indicating that it is an X-linked recessive inheritance. So actually this is a pedigree analysis for hemophilia as we will talk later about it, as a representative example of X-linked recessive inheritance. So you can see here, it starts with Queen of Victoria that married Prince Albert. And you can see all the generation, and you can understand from the key here, which is affected, which is male with hemophilia, and which is possible carrier, and which is confirmed carrier. Okay, so that's the normal, or whatever. This is the pedigree analysis. What is interesting here, what's interesting, that you can see here, this family here represent actually the British royal family. And these individuals, the British royal family came from Queen Victoria. And as well, these individuals here and here represent actually the descendants of the current German royal family and the Russian royal family as well. While here, the descendant of this branch from Patricia represent the Spanish royal family. So actually, Queen Victoria, that's why it is she is termed the mother of Europe, because actually all its descendants gave to the royal family of the Spanish and Russia and Germany and British. Okay. So these modern genetic studies are now using genetic markers present in the DNA itself rather than the phenotype, because actually it is more precise to examine the genetic markers rather than the phenotype. So let's take for an example how they can study genotypic by molecular analysis a genotype rather than the phenotype. So there is an expression called single nucleotide polymorphism, and the human genome include one, that's mean the DNA, our DNA, usually include one SNP per almost 3,000 base pair. So that's mean from the region of 500 to 3,000 base pair, there is one SNP. What's one SNP? It's a single nucleotide polymorphism, in which you can see here, that's the normal DNA molecules. Different individuals could have different version in one nucleotide in any of the genes. And this is called SNP, or single nucleotide polymorphism. So actually it's random, and you can have a location on your DNA, even the same gene of insulin, for instance, it could be one nucleotide that is changed, and in another individual, another nucleotide is changed. So you have actually, you can see 4 different possibilities for each nucleotide that generate a different version of the genes. For example, an example of DNA polymorphism is the simple sequence repeat polymorphism. So actually it is a sequence of nucleotide that present in our DNA, and this sequence of nucleotide exists in our DNA, and they can be detected by size. Let's see more details about this one. So actually this SSRB actually have 6 alleles. So actually it's a gene, okay? Why 6 alleles? Because as we said, there is SMB between these gene, so you have 6 versions of these genes, so that's why you have 6 alleles. These 6 alleles is different in size. As you know, normally we have 2 alleles, a dominant allele and a recessive allele. But in this case, SSRB have 6 alleles from A1 to A6. So the difference of size of this gene can be detected by electrophoresis. If you remember, as I said in the first of the lecture, there is a difference in size between the round phenotype of the B and the mutant phenotype of the B. So you can see that the majority of individuals, they have been found that they are heterozygous for the 2 different alleles. And when we have the pedigree analysis, you can see that these 2 individual mates, they will give these individuals, and these 2 individual mates will give these 2 individuals. When this one mates with this one, they will have all the possible individuals you can see as a combination between the different alleles they have. When they do the analysis on the gel, you can see that A4, for instance, A4, A6, you have 2 bands here, and this is the size of 4 and 6. A4 and A5, and the A5 parent, when you do the molecular analysis, you can see 4 and 5. So basically, it's better to check the alleles by the molecular method, then got confused by the phenotype, especially with this gene, the SSRB. You can easily see the phenotype of it upon these 6 different alleles. So you can detect this phenotype, this genotype by the molecular method to know which allele is present. For instance, you have allele A1, A3. It will migrate 1 here, 3 here. So if you need to check if the parent, which of the parent have the allele A3, you run the agarose for the parent 1 and parent 2, and of course you will see that allele 3 is present in this parent, is not present in this parent. Okay? So this slide basically shows that you can detect the genotype by the electrophoresis analysis to understand which allele come from which parent by comparing the pattern of the separation of the DNA sequence on the gel electrophoresis. For the 5th topic now, we'll talk about non-Mendelian inheritance. So actually, what is non-Mendelian inheritance? So actually, it is the inheritance that doesn't follow either the first law or the second law of Mendel. So there is a number of examples like incomplete dominance in which neither of the 2 alleles is fully dominant nor recessive, and the resulted phenotype in the progeny, actually it is both traits are blended together, and we'll talk about our example, the snapdragon's plant. Well, the second case is codominance. The famous example of it is ABO blood grouping, in which both of the alleles express in the phenotype. The third, the non-Mendelian inheritance, appears in the multiple alleles. As well, ABO blood groups and SSRB, the one we just said, is an example of a multiple allele. That means their inheritance is totally different than Mendel inheritance. The 4th type of non-Mendel inheritance is epistasis, in which one gene masks or modify the phenotype of the second gene. And in this case, we'll talk about the flower example, the flower coloration in bees. The 5th non-Mendelian inheritance is the polygenic traits. And actually, the polygenic traits, the famous example that the trait is affected by more than 1 gene, not allele. So it's totally different. More than 1 gene. The famous example is height and skin color and the eye color as well. The last subtopic in non-Mendelian inheritance is the 6 linked traits. And of course, the 6 linked trait, it is the trait that is transmitted to the generation, and it is dependent on the sex of the parent. The famous example is hemophilia and baldness as well. So let's see the incomplete dominant. In this case, it was found that the mating of a red dragonfly or snapdragons with an ivory dragonfly will result in a generation that have a mix of the 2 phenotypes, which are around a pink flower, and this is a totally new phenotype. Upon the self-fertilization of this F1 generation, it gave rise to, upon self-fertilization, it gave rise to 3 different phenotypes: the red and the ivory and the pink. Okay, so what does this mean? This means that you have an allele, which is I capital allele, is responsible actually for the amount of the red pigment. Once it is homozygous dominant, the flower is purely red. And once it is homozygous recessive, there is no red pigment, so it appears white or ivory coloration. And in case of the heterozygous, I capital I small, the flower is a pink. Why? Because actually the dominant allele produce half the amount of the pigment, so it's a kind of a dilution for the color, and that's why it appears pink. So one would expect the ratio, according to Mendel, to be 3:1. So probably you will have 3 red to 1 white or ivory, but in this case you have 1 to 2 to 1, 1 red to 2 pink to 1. So that means there is an incomplete dominance. Although I, big I, is dominant, but it is incomplete because it, in case of heterozygous, it produces half the amount of the pigment. So that's why it gave the pinkish color. The second example of Mendelian inheritance, actually, it represented the codominance and the multiple alleles. And the famous example of that, the blood grouping. So normally the blood group is expressed by 3 different alleles, either IA, IB, or IO. So these are 3 different alleles. That's why it is an example of a multiple alleles. And as well, it represents an example of a codominance. Why codominance? Because in blood groups of AB, you expect that once there is IA, IB, you need to think, so which is dominant over which? Actually, there is no dominance over, no allele is dominant over the other because there is a codominance. That's why you will have a new blood group, totally different blood group, which is called AB. So this AB group represent a codominance representation of these 2 alleles. And of course, you know that O is universal donor, while AB is universal acceptor, and each of the blood group can donate to each other. And this actually based due to the fact that blood type A have the antigen A, while blood type B have the antigen B. And once there are, for AB, on its surface, it have both the antigen, so that's why the AB, for the O have no antigen at all, so that's why it can donate for everybody. But AB have the both type of antigen, that's why it can accept from all over the other blood groups, and no agglutination occur. Okay, so again, the blood grouping is an example of a multiple alleles. Why multiple alleles? Because it is controlled by 3 alleles here, not 2 as usual as Mendel. The 3 alleles are IA, IB, IO. So once IA, it's a dominant character, it will give a blood group. IA, IO, it behaves as Mendel because IO is dominant over O, so it will give a blood group A. The same for B. But the problem here, the codominance here is represented by the 2 alleles IA, IB. You expect again here that one of them will be dominant over the other, but this is not the case. And you'll have another blood group which is called AB blood group. Continuing to the multiple allele, again, so the 3 alleles of AB group is one example, and these are the 3 alleles. The 6 alleles of SSRB we have just said is a second example because they have from A1 to A6 alleles. These multiple alleles are relatively common in natural population and can be detected, as we said, by molecular methods. In a DNA of a gene, each nucleotide, of course, can be A, T, G, or C. So a gene of n nucleotide, whatever number of nucleotide, can theoretically be mutated at any position to any of the 3 other nucleotides. So A, for example, A can be mutated to T or G or C. So the number of possible single nucleotide difference in a genes, keep in mind, we are talking now that these changes in this single nucleotide in a gene will result to a different allele of the gene. So we are here calculating what is the possible number of different allele from a single gene. So you can see that if the number of the possible single nucleotide difference in a gene of n nucleotide is 3 times n. So if n is like 500 nucleotide, so there are 5,000 nucleotide, multiply by 3, so you will have 15,000 different alleles. That's theoretically. And of course, here you are not counting the possibility that 2 nucleotide is substituted. It's only, you are only counting the possibility of 1 nucleotide is changed to another nucleotide. Okay? The next example of non-Mendelian inheritance is epistasis. So what's the epistasis? Epistasis actually, as we said, when one gene masks or modifies the phenotype of a second gene. As you know, the typical ratio, F2 ratio in case of Mendel's second law is 9:3:1. In this case of epistasis, this ratio is not observed. So there are gene interaction that disrupt the normal Mendelian ratio, and in this case, we are dealing with the epistasis. So one type of this epistasis is illustrated in the interaction of two genes, actually represented by two alleles, C capital C small, P capital P small. And these allele pairs affected the flower coloration in bees. So again, this flower coloration is controlled with these two genes. So these genes encode enzymes used to synthesize an important pigment, anthocyanin pigment, which requires this pigment to be probably formed by the flower to give the pigment. It requires the presence of at least the C capital, P capital. So it requires at least the presence of C capital, P capital, irrespective of the other allele for each gene. So it requires one wild-type dominant allele for each gene, okay? So you can see here, in this violet color or pink color, there is formation of pigment, the formation of the pigment of the require, as we said, C dominant, P dominant allele of the genes. With this type of epistasis, the dihybrid cross, if it goes with Mendel's law, probably it will give ratio 9:3:1. But scientists have found that the ratio have been modified to 9 purple and 7 white. So what's happening? Why it's 9:7? This means there is something wrong on the dominance recessive interaction between these genes. So let's see here. We have here homozygous mutant B small, B small. You can see here the parents actually looking white. Why looking white, not pink? Because as we said, the violet color needs one dominant from each gene to have the violet color. So in this case, this dominant, homozygous dominant for C and homozygous recessive for P, so this one lacks the dominant for P. So that's why it's white. Same as here. This flower is white because it lacks the dominant C, So once mating occurs, it gives rise to the violet, which now it have one dominant allele for each gene, and which it is required, these two dominant alleles for each gene is required for the production of the pigment for the violet color. So what's happening normally, if it goes according to Mendel, if you've done self-fertilization of this flower, it must give a ratio of 9:3:1. But they found that it behaves normally, and the gametes normally behave as we have just said. But the offspring appear, 9 of them appear purple and 7 of them appear white. Why? Because you can see in the white, it lacks one dominant allele in one of the two genes. And for the violets, all of them to be violet, it needs at least one dominant allele for each gene. So again, so in this epistasis, actually, for the pigment to appear, it needs one dominant allele for each genes. The absence of one of any of the dominant allele of any of the two genes will give rise to not enough pigment, so the flower will be white. The next example of non-Mendelian inheritance is the polygenic traits. For polygenic traits, actually it is trait which is controlled by multiple genes instead of just one. The genes that control these multiple genes may be actually located near each other, so all these multiple genes may be side by side on the DNA, or maybe on separate chromosomes. But anyway, they control a certain trait. So polygenic traits are represented by a range of continuous variation. So you don't have a definitive phenotype, but you have a continuous variation. And this phenotype, or a person phenotype, is a result of interaction of all these multiple genes. And also as well, they're influenced by the environment, like the sun tanning. The skin color, actually, the skin color is controlled by polygenic. It's a polygenic gene, a polygenic trait. And it can be affected by the sun as well. So it's not only the interaction of these multiple genes that lead to the polygenic trait, but it as well the influencing of the environment. So the major example here, there are two major, the eye color. Actually, there are two major human eye color genes that is responsible for the color of the eye. These two genes, and at least there are 13 other genes play a major role in the eye color. Actually, the color part of the personal eye is in the iris. So a person's eye color is determined by the pigmentation of their iris, but also by the way the cells in their iris scatter light from outside. So that's the environmental effect. So again, the eye color is controlled by two major genes and other 13 other genes. And basically the pigmentation appear in the iris of the eye. Although there are two genes and the 13 other genes, but still some environmental factors like the way the iris scatter light. So as with the skin color, it's the same case with skin color. Eye color is affected by the presence of melanin. A lot of melanin will make the iris dark. People with brown eyes have a lot of melanin, while people with blue eyes have low melanin in their iris. While green eyes are caused by multiple factors. They are as a result of light brown iris, that's mean a very few amounts of melanin, as well combined with the blue tone given by light scattering. Okay, so here we show that these polygenic traits, although it can be affected by multiple genes, here, for example, like 13 different genes for the color of eye, there are as well other environmental factors like the scattering for the eye, like the scattering of the light, for the skin, like the exposure to the UV light, which may affect in this trait. Let's see here an example. So you can see here the difference of the eye color and the difference of the skin as well. So generally, any polygenetic trait, it is distributed in the form of a bell shape. So we have here, there are common cases that represent most of the population, and there are rare cases at the extremes of the population. For instance, if we are talking about the height, so you have the rare cases. In this case, you have short, very short people, that's a rare case, and very, very long people, that's a rare case as well. And you have most of the population is around the average height. So that's why we are saying that the polygenic traits always give rise to a range of continuous phenotypes. Not blue, not green, no, it's a range of height, range of colors. Even the skin, there is a range of the skin, range of color of the skin. There are some polygenic diseases that result from a polygenic effect. So this genetic disorder caused by polygenes. One of the famous examples which can be affected by a polygenes is type 2 diabetes. So according to recent findings, there are more than 36 genes, and each of these genes contribute to the increased risk of the developing type 2 diabetes. The last topic in non-Mendelian inheritance is sex-linked genes. And of course, you know, for sex-linked, you have X chromosome and Y chromosome. And these traits are carried only on X and Y chromosome. In humans, the Y chromosomes have around 55 genes, and the X chromosomes have around 900 genes. And of course, you can see that a Y chromosome is very short compared to the X chromosome. And of course, the genetic of these genetic is called sex-linked, and sometimes they say X-linked because most of the gene is carried on the X chromosome. And due to the mutation in the genes on the X chromosome, any phenotype usually only seen in males. In mammals, the females, of course, are XX, while the males are heterozygous XY, and usually it's called hemizygous. So what's the meaning of hemizygous? Hemizygous means that the Y usually lack the allele which is present in the X chromosome. And usually Y chromosome even have a separate set of genes that is totally different than the X chromosome. So we have sex-linked inheritance. This sex-linked inheritance can be divided into two broad categories, either X-linked inheritance or Y-linked inheritance. For an X-linked dominant trait, for the character to appear, the female had to have the two as well. It's like Mendelian, XA or XA, either homozygous dominant or homozygous or heterozygous for the dominant trait to appear. For males, it is enough one allele to show the trait if it's dominant. As well, the female, to show the recessive trait, it needs to be homozygous recessive. So basically, it follows the same pattern of Mendelian, but the exception here, it follows the same pattern of Mendelian, but it is linked to the sex chromosome. Hence, males, since they are only having one X chromosome, they are much likely to express the negative allele. So for instance, if the disease is caused by a recessive allele, normally this recessive allele, if it's existing in females, probably it will be masked by the dominant allele. But if this recessive allele is present on the Y chromosome, it will appear because it will not be masked because there is no corresponding allele in the other chromosome. So let's take examples for the sex-linked trait. First example is the Fragile X syndrome. In this Fragile X syndrome, you have females that will be affected if their father have the disease, but the males won't be. Boys and girls will have 50% chance of inheriting the disease if their mother is heterozygous. While in case of X-linked recessive disorder like hemophilia, the males are more likely to develop the trait than females because girls need the two copies, homozygous recessive, of this allele for the trait to be expressed, while boys, as I just said, only need one copy. If a female is heterozygous, this means she is a carrier for the allele, then neither she nor any of her daughters will get the trait. While the third example is the Y-linked inheritance, and usually called Hollandric inheritance, and the famous example for that is the excessive hair growth in the ear. And it occurs only in males, and the trait is always passed from father to all his sons. And you can see here how the X-linked dominant inheritance is moved or transferred to the generation, and as well X-linked recessive inheritance. So you can see if the mother is affected here, if the mother is affected, you will have 50% chance of the whole progeny to be affected, okay? Irrespective, male or female, since the mutant allele is existing on X chromosome. So the X chromosome could pass to a male or could pass to a female. That's in case in X-linked dominant. While for X-linked recessive, here the affected, there is no affected parent, but there is a carrier mother that have the mutant one. So this a carrier mother, you can see that there is 25%, you can see only one of the four is affected, while the other, the female, it passes, it have a daughter that is carrier, and both of the other are affected. So the severity here is more if the case is X-linked dominant. While for the Y-linked, the story is different. If the disease is on the Y or the mutant allele on the Y chromosome, all the sons is affected straight away. And the female is always unaffected because they will never receive the Y chromosome. Okay, let's see here. What is the number in the circle? Can you tell me what is the number in the circle? So what number you can see here? Actually, it is a 74, okay? So it is a red-green color blindness. It's common inherited trait in humans. So it's the X-linked recessive disorder. So about 1 in 10 men have some form of color blindness. Very few women are colorblind. That's why females have a range of color in their eye when they buy their stuff, which is more, the spectrum is more than the males, if you understand what I mean. So females are red-green colorblind only if both of their X chromosome carry the defective genes, whereas males are colorblind if their single X chromosomes carries a defective gene. So you can see here, so here you can see actually this is a pedigree, and you can understand, and you can see here how the trait is moved from. You can see that the trait is represented in all males. Here you can see in the offspring here. So that's mean it's a kind. It gives some indication that this is a sex-linked disease, and actually this is actually the pedigree of color blindness. Okay, let's see what is the obstacles and the challenges to study the human genetics. Before molecular method, there were many practical obstacles to study the human genetics. So basically, you can't experiment human beings. Most of the traits showing simple Mendelian inheritance were associated with genetic disease, and they presented special challenges. What are the challenges? Actually, that most of the genes that cause this Mendelian genetic disease are rare. So they are observed only in a small number of families. As well as many genes for this Mendelian disease are recessive, so they are not detected in heterozygous genotypes. The number of offspring per human family is relatively small, so segregation cannot usually be detected in a single sibling. So in other words, if you have a percentage 3:1 for disease to appear, that means the homozygous recessive to appear. That doesn't mean if the family have 4 children, for sure one of them will have the condition. Okay, and maybe the family give rise have only 1 child. So that's why it is very hard before to study the genetic of human, because all the representative genotypes in the parents and the offspring will not represent it. The last challenge is the human geneticist cannot perform Testcross or Backcrosses or other experimental matings on humans. Okay, so these are the major challenges of studying genetics on human. The last topic we are talking about today is the genetic today. And actually, genetic today is not dealing with classical genetics. It's dealing with recombinant DNA technology, genetic modified organism, GMOs, whole genome sequencing, and genome-wide association studies, and gene therapy. We will not talk in details in this one, only the headings, but you need to know that recombinant DNA technology, the famous example of this one is the production of recombinant insulin, which is being used now in the treatment of diabetes. While for the genetically modified organisms, one of the famous genetically modified organism is the yellow pepper, yellow sweet pepper and red sweet pepper, because normally the wild type is green, not yellow and red. And for the whole genome sequencing now, they do whole sequencing for organisms to identify the nucleotides and the genes. And for the genome-wide association, actually, they relate any mutation to a certain and try to associate this mutation with a certain condition of a disease to try to predict or correlate if someone have this mutation or this allele, it might in future show this kind of disease. So for GWS, genome-wide association studies, normally it's a kind of a correlation studies to predict the possibility of a certain disease to appear or not. And of course, the last thing is the gene therapy, and it's a big aim for trying to cure disease by replacing the defective genes with a wild type. So that's mean replacing the mutant gene with a wild type gene again. and there are a lot of experiment is going on in this field. And that concludes our lecture for today. I'm waiting for your questions.